The cerebral cortex is the surface of the brain. And we talked about this a little bit in the introduction, gross anatomy of the brain. We pointed out all of the different, the sulci, which are the great big divots in between, and the gyri, which are kind of the bumps. This is your neocortex. Your neocortex means new cortex, okay? And it's unique to mammals. So reptiles don't have it, birds don't have it, fish don't have it. It's a mammal thing. So be proud that you're a mammal and you have neocortex. The surface area of the cortex, when we talked about gross anatomy, I mentioned that your brain is too big for your head. And the fact is that, and that's why it's in all these folds, these sulci and gyri. So if you could take your cortex and flatten it out, it would be about 2.5 square feet. So about the size of a newspaper. And so if you think about that, you think about taking a newspaper and kind of turning it into a balloon, that's how big your head would have to be if we didn't have sulci and gyri. The cortex is about 80% the mass of your brain, and it's where you think. It is really where you, as a person, exist in your nervous system. So everything that's unique to you is because of your cortex and is a property of your cortex. It's influenced greatly by all of the sensory information that's coming in from the outside, everything you see and hear and smell and feel, and internal stuff, the internal workings of your own body, the things that that your central nervous system can sense, and you can kind of self-generate. You think your own thoughts. So there are multiple levels of stimulation that are being integrated within the central nervous system, and most of it is going to be integrated in the cerebral cortex. So as we said, neocortex is new cortex, and it's 6 layers. And we can see some of those layers here. In this cartoon, we can see there's layers 1 through 6. There's an outer kind of molecular layer. There are layers 3 and 5, which are these great big pyramidal cell layers. And then there's layers 2 and 4 and 6, which are receptive layers. We'll talk a little bit more about those when we talk about cytoarchitecture. Archicortex or paleocortex, think about archaeology or the Paleolithic period. Those are both terms that mean old. And so there is old cortex in your brain left over from, evolutionarily speaking. And one area of this is found in the hippocampus. Old cortex only has 3 layers. So again, layers 1 through 6 are superficial to deep. So layer 1 is out here. Layer 6 is here. And there are basically layers that send information and layers that receive information. The layers that send information are these layers that have the great big cell bodies. Because if you think about it, if you're sending information all the way from your cortex, think about the motor system, all the way down maybe to the bottom of your spinal cord, you've got a really long axon to maintain. And if you have a really long axon to maintain, you've got to have a really big cell body to maintain that axon. If you are a layer that receives, so you're getting all kinds of information, you want to have a whole bunch of different neurons in there. So areas like this would receive, and area 4 is big into receiving. And you have big dendritic arbors. So lots and lots of areas for synapses to take place. I'm going to erase and turn my drawing tool back on. What I also want you to note is it depends on how you look at the cortex. So this image is a Golgi stain. Is a kind of an artist's rendition of a Golgi stain. Golgi stains were discovered kind of accidentally by dumping a chunk of the brain into some heavy metals. We don't know exactly how it works. Still to this day, we don't know how it works. But the bottom line is that the heavy metals will fill up the entire cell body, and it's axons and dendrites. It doesn't fill up every cell, because if it did, it would be just one big black chunk of tissue. But it does fill up the odd neuron here and there. Nobody knows why some neurons are filled and some aren't, but it works really well to see the entire architecture of a single cell. The middle panel is a Nissl stain. Nissl substance is big stacks of rough endoplasmic reticulum, which are responsible for making proteins. And so neurons make a lot of proteins. They make neurotransmitters, they make all kinds of proteins to put into their channels and their receptors. And so this stain stains mostly the cell body. So you don't see a lot of processes, but you see the shape of the cell body. And it stains almost all of them. So you have a different look if you use a Nissl stain versus a Golgi stain. In this last panel, we've stained for myelin. And myelin is that lipid-rich coating that surrounds all of the axons to keep them to keep crosstalk from happening. So if you have a signal going down one wire, you want it going only down that wire and not down another one. And so you have to keep them insulated. It's just like the insulation on the wires in your house. So two axons side by side have to be insulated from one another, and that is what myelin does. And if you stain for the myelin, what you notice are these columns of cells separated by these bands of fibers that are going along this, kind of making these columns within the cortex. Well, it turns out that these columns are kind of the functional unit of the cortex. So neurons within a column are working together, and then they talk to the neurons in the next column, or the next column this way. But these guys are all working together. In the next section, we'll talk about more of the cytoarchitectonics and what that means with regard to function.